everyone. So welcome to the second EMSL LEARN webinar, where today we're going to be discussing how you can visualize root architecture for your research projects. Next month's topic for EMSL LEARN will be using cell-free expression for protein analysis. We hope to see you there too. Also, make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. The links will be put into the chat window. So for today, you're going to be hearing from Amir Ahkami on imaging and analysis of root system architecture with an automated non-invasive phenotyping system. You'll hear from Tamas Varga on X-ray computed tomography resources at EMSL for root imaging. And you'll hear from Pubudu Handakumbura on visualizing the hidden half of plants. And without further ado, Amir, take it away.
All right. Thank you, Linda. And hello, everyone. And thank you very much for joining us for today's EMSL Webinar and Learn Series. So I'm Amir Ahkami. I'm a staff scientist at EMSL. And by training, I'm a plant biologist and currently acting as a science lead for plant and ecosystem phenotyping integrated research platform. So at this part of the webinar, I'm going to talk about imaging and analysis of root system architecture using an automated non-invasive root imaging. So as you may know, there are many different types of techniques that enable us to do root phenotyping and root imaging. Some of them are based on artificial root environment, and some are based on natural soil condition. So depending on the goals of the project and available funds and instrumentations, basically any of these techniques can be used for root phenotyping. Of course, all of this comes with its own pros and cons. And having access to all is ideal, and because each of these techniques could basically complement each other. But here, I'm mainly going to focus on the use of transparent growth medium. And particularly, I'm going to focus on using gallium gum for root phenotyping. I will also talk about our recent efforts on making and using transparent soil for root imaging. So overall, there are two major components of root phenotyping techniques. Which actually one of them is adding automation to the facility of root phenotyping. And the other one is increasing throughput. So these are the major two challenges and components for any root phenotyping facility, which we try to address both. Next slide, please. Okay. So as part of our effort to expand our root phenotyping capabilities, we recently developed a new system called NARI at EMSL, or Non-Invasive Automated Root Imager. So actually, Tom Witzma mainly built this system for us at PNNL. So with this platform, multiple digital images are generated automatically while plants are grown in a glass cylinder jar containing the transparent medium, which is made with gallium gum. And gallium gum, as you probably know, is a water-soluble polysaccharide. So the EMSL NARI system consists of a machine vision camera, as you see in this picture, optical correction tank, NIR LED backlight, and turntable mounted on a custom-built table. So to image the plant roots in this system, a cylinder which is filled with filled with the gel is submerged in a water tank, and then imaged as the root system and plant is rotating in the turntable. So it rotates through 360 degrees and collecting 2D images. Next slide, please. So to optimize the system, we established growing brachypodium as a model grass, healthy brachypodium plants in our gel-based system, and collected images. And we aim to collect more than 90% of the root system architecture, which in the case of brachypodium it reveals third-order branch roots with a diameter of about 85 micrometer. So the number of factors that needs to be optimized with this system, for example, percentage of the gel is very important. It needs to be optimized for each species. Depth of the seed in the growth medium is also playing an important role. And of course, contamination. So there are many ways that we really need to deal with to reduce contamination rate. Use sterile labware, autoclaving very carefully, autoclave seeds, sterile seeds. And of course, as you do this more and more, then you will also you'll able to use some kind of common sense approach. And this is kind of our control in our lab right now. So we eventually made it in a way that we made the contamination rate in a very low level. And then we also optimized the imaging platform by growing at least 8 brachypodium plants as biological replicates for more consistent data. So some additional technical adjustments may be needed. For example, adjusting the aperture of the lens of the camera, so which could impact the depth of view. So as you see here, the NARI system generate multiple images representing 360 degrees in 10 minutes per sample. So we can have, for example, 36 images, as you see here. If you adjust the camera in a way that it takes images in every 10 degrees, but you can also get 72 images if you adjust the camera to take images in every 5 degrees. And then we have all images for one plant system as a stack, and then you can use that for image analysis. So once images are collected, then they are optimized for by thresholding, which I mean selecting a pixel above a given brightness to segment the image into foreground value and background values in a way that all or nearly all of the root system architecture are visible. Then it's ready for further root trait extraction and identification of root types. Next slide, please. So as I said, the next step is data analysis, which includes image processing and root trait extraction. So for that, we're mainly using freely available software packages and a directory of available root image analysis tools, and their information are available on the quantitativeplant.org website. We recently used a software package called Rhizo Vision Explorer, which is recently developed, and we collaborate with the developer, Larry York. And we also use our package for when we extract root traits from Rhizo Vision Explorer, then we use our package for further statistical analysis. And with that, we were able to extract more than 20 root parameters, including root width, depth, length, solidity, perimeter, number of root tips, and many more. Next slide. Yes, and then here is the fun part starts. We have lots of data, and this is just some of the results from a test experiment, with 8 brachypodium plants that imaged for 5 days. Just to show you a sense of what's going on, I just show 4 examples of 4 parameters: diameter, convex area, depth, surface area, which, as you see, they're changing during time. And we also included a random forest variable importance graph, as you see in this slide, which this actually can be used to help figure out which of these parameters is most important in influencing difference between the different time points here. Okay, so there are some advantages and applications for our system. Of course, this is provide a 3D growth environment and also a homogeneous conditions. Because it's gel system, there are some physical constraints for root growth. It is automated, and I would call it as a semi-high throughput at this point. As I said, it is it takes about 10 minutes to image one individual plant. So we can handle 40 to 50 plants to be imaged per day. The applications, so we can use these to derive and study, to derive root traits and study root morphology under nutrient deficiency, under salinity stress, or even during plant microbe interactions. And then the data derived from NARI could relate genomic information to root phenotype. So if establishing a link between root system architecture and genotype is a major goal of a study, then it usually requires the measurement of root phenotypes, which we can provide that through automatic analysis of digital images that we can take by NARI system. Next slide, please. Okay, so for example, we just work with an EMSL user, Patricia Akebora from Washington State University, to study early root architecture of rhizoctonia-resistant and susceptible wheat plant. So in her preliminary studies, Pat shows early root growth rate has been negatively correlated to genetic resistance against the fungal pathogen rhizoctonia. And that study was conducted in Petri plate assays. But now we're employing NARI for root phenotyping, which will actually help us to investigate whether the genetic resistance in wheat is correlated with early root developmental traits. So the ultimate goal here is to predict disease resistance among unknown genotypes and possibly modulate genetic resistance by selecting for specific early root morphological phenotypes. So here are some data. We recently generated these data, some data under this user project. I really don't have a plan to go to details and explain everything. We really need more time to dig into this data and extract more meaningful information out of that. But just to give you a sense that what can be done with NARI, I share a small portion of data with you here in this slide. So we extracted more than 20 root traits using RhizoVision Explorer software package and did a more comprehensive data analysis with R. Just highlighting a few of the parameters here. For example, we measured a parameter called solidity, so which is actually the ratio of network area and the convex area. So by determining this, we can have a sense of how the root biomass is distributed within the network shape. So for example, if a genotype shows lower solidity, it means that the roots are ranging widely and deeply with large gaps between them. But if a genotype shows a higher solidity, it means that the roots are growing densely together. Or another parameter called holes here. So holes are kind of background components between roots in a segmented image. And this parameter is indicative of root branching and complexity. So which, as you see, these two genotypes are significantly different regarding this parameter. But for example, there are no significant changes regarding their volume. So overall, these traits distinguish features of root systems for each genotype and complement one another in important ways. So for, like many other facilities and capabilities, there are challenges with this system as well and some limitations. We really need to have high number of replicates when we work with NARI, just to provide more accurate data and for consistency between growing roots. So we need high number of replicates for sure. The other thing is that although multiple 2D images and their analysis provide a good estimate of some root system architecture parameters, ideally 3D images are needed to provide trait measurements more accurately. So here we use multiple images in 2D as a proxy for 3D reconstruction. because 3D, as a proxy for 3D representation, because the 3D reconstruction is not possible with the current version of our setup. But there's a solution for that, and we're working on it. So we work with other computational scientists to implement an algorithm that generates 3D reconstructions from rotational image series that we take with NARI. So the other challenge with this system is that it is artificial environment anyway. So, and it's hard to mimic the real environment. Of course, there are lots of benefits of using this. And as I said, initially, all of these techniques can complement each other, but it is artificial. But there are some ways to at least get closer to the real environment. And one way to do that is moving from gel-based system to transparent soil. I think in my very last in-person conference about 2 years ago, so in Arizona, I had a chance to get to know this paper published in PNAS, where actually authors describe a porous medium as a hydrogel-based transparent soil that allow for the imaging of root system in vivo. So when making transparent soil, it is important to develop media that have air-filled connected pores because that medium should show several relevant characteristics of soil. And as you see in this picture, so they show that the root structure developed in transparent soil is visibly more similar to that developed in field soil compared to the one developed in hydroponics. So based on that, we decided to make transparent soil in EMSL. We made some modification to the original protocol, and we did it. So briefly, hydrogel beads are produced by dropping a solution of gellan gum and alginate into a stirred solution of magnesium chloride. And then we tried to use this transparent soil with NARI for automated root imaging. Next slide, please. Yeah, as you see here, we grow some test plants in the transparent soil. And then, so we actually added the nutrient solution to that. So this nutrient solution here, this treatment is comparable to a rainfall or watering, because that makes this medium sufficiently transparent to allow imaging. If we move forward with the slides, yeah, as you see here, so it makes it transparent for imaging. And then we also image that plant with NARI and process that, did thresholding, and so the root system is completely clear for further analysis. So we confirmed that growing plants in transparent soil here produce roots more like the ones grown in real soil. And it also provides high transparency and a good mechanical stability for us. And it is made relatively easy and with low cost. So there are potentials here to invest more time and employ transparent soil for automated root imaging in our future studies. Okay, now I really would like to thank the great team that I'm working with, and of course, EMSL management for their support during this work. And I should also thank Philip Benfi at Duke University, who actually for their very great advice on making NARI system at EMSL, and particularly Christoph and his lab members at the Donald Danforth Plant Center. So they helped us a lot during the development process. Guys, stop here, and I'm happy to pass the stage to Tamas. Thank you.
Hi, everyone. Thank you for joining. I'm Tamas Varga, a scientist at EMSL, and I will talk about X-ray computed tomography in general, and also as we use it for root imaging. So this is what we want to achieve. We like to image the plant in its native environment, the soil, and then extract its 3D representation and learn about it. So how can we do this? We use X-ray computed tomography for XCT. And this technique is a non-destructive X-ray method. Next, please, next slide. XCT is a non-destructive X-ray technique to image and visualize the internal microstructure of objects. Here you can see a general concept for most lab X-ray systems. We put the sample in between an X-ray source and the detector on a rotating stage. And as shown here on this example of a sediment column, as the sample rotates a full 360 degrees, we collect thousands of radiographs on the detector, which is then computationally reconstructed into a 3D data set. And in this 3D data, we differentiate the components based on their X-ray absorption. The components that have more electrons, or they are higher density, they absorb more of the X-rays, while the components that are less dense transmit more X-rays. And this difference will show up on the detected images as a difference of in intensity or grayscale color. Next, please. At EMSL, we try to cover as wide resolution range as possible between the extremes of a field scale and the molecular scale. So we have this Nikon CT system, which on the left covers the kind of the centimeter to down to 10 micron resolution range. It's applicable to larger soil cores to detect larger pores. It's perfect for imaging whole plants, the whole root architecture. While in the middle we have the micro CT. There is a HeliScan micro CT at PNNL that we can also access, but in the future we would like to acquire one for EMSL. This covers hundreds of microns down to sub-micron resolution, which is perfect for imaging soil aggregates, individual particles, detecting micro, even nanopores, and fine root hair to learn more about the rhizosphere. And then at the other end, we have the nano CT, which will become available for EMSL users later this year. This will be my X-ray microscope geared towards biological samples, a soft X-ray system that can cover down from tens of microns down to 20 nanometer resolution, perfect for imaging cells, fungi in 3 dimensions. The system I am responsible for at EMSL is this large cabinet Nikon XTH 320-225 system, which, as you can see in the image, there is a large soil core on the stage, and this system has 3 different X-ray source options, as listed here. We mostly use the 225 kilovolt fixed target, which gives us the highest resolution, but we also have an option for higher high penetration X-rays for dense large objects with the 320 kV. We have different wavelengths or X-ray energy options by rotating or changing the reflection target to different metal surface, and we can also use different filters to further monochromatize the beam. Currently, we are running with the tungsten target, but we can change it to the other metals. The detector is a 2000 by 2000 pixel large panel detector, and the sample can be moved between the X-ray source and the detector to achieve different magnifications. And I'll tell you more about this for the spatial resolution. Data collection can vary from minutes to hours, depending on the sample and the amount of information we need. Most cases we run 1 to 2 hour scans on most samples. And so we have this cone beam configuration and the related reconstruction algorithm we're using. Next slide, please. A few words about resolution. So we we can talk about spatial resolution and phase resolution or phase contrast. The spatial resolution depends on the sample size on one hand. On the image on the left, you can see that if you want to capture a smaller object in the cone beam, we can move closer to the X-ray source, and we can achieve larger magnification and higher resolution on the detector screen. But if we have a large specimen, and if we want to capture the whole volume, we need to move away from the source, and thereby lowering our resolution. Typically, we say that the 1/1000th of the object diameter is typically the voxel size that we achieve. The spatial resolution also depends on the X-ray power, because the focal spot size changes with power. So the best resolution I usually quote as 10 microns for the system it can only be achieved at limited kilovolt and microamp settings. If you go to high power, then your focal spot size becomes bigger, and that compromises the resolution. And for the phase resolution, we can change the X-ray energy, and if we have a way to change the densities of one of the components, then we can play with the phase contrast. This slide shows a few examples for how the resolution depends on sample size. In the middle, we have this potted plant, which was about a 3-inch diameter pot. If you want to image the whole volume, then you get about 130 micron resolution. If you want to image a gallon-sized sample, like bucket on the right, you achieve about 250 micron resolution. And on the lower end, with a 10 millimeter diameter cylinder, you can get about 30 micron resolution. And I know that in this table, I quote the resolution as double the voxel size, since typically we need at least 2 voxels, if not more, to recognize or resolve a feature in CT images. So to be conservative, we say about at least double the voxel size. Next slide. So here are some typical examples of samples at EMSL. As I mentioned, we get a computerized model in 3D of the objects that we scan, and the root architecture visualization is one goal that we often have. Once we achieve this segmented root image, we can study the root structure, map it to drought, varying CO2, temperature conditions. Basically, we can study how the plant responds to environmental perturbations. And then from the segmented image, we can measure the root volume, root surface area, diameter, etc. On the right, in this example, is a soil project that was published in 2017. So for soil samples, we most often characterize the porosity of the soil. This was a small soil core, and I'm only showing a cropped volume of it, or part of it. By visualizing the porosity, we can learn about how, for example, the wetting of the soil depend on the pore structure and flow direction. We can also characterize pore network connectivity, which has a bearing on the transport flow of water or transport of nutrients in the soil. Next slide. Now, just to talk a little bit about how we can distinguish different components. This is an example of a multi-component composite where we have 4 different components. And on the right, you see a red rod appearing, which basically represents the lowest density component. From left to right, we will go from lower density to higher density. So this is air visualized. If you go next, the green phase is the polymer in this case, somewhat higher density. As you move further to the right, you get the cement matrix. And then we also have this highest density mineral inclusions, probably metal containing oxides in this volume. And so basically these 4 components make up the whole volume. And once you separate them this way, you can visualize them and do volumetric measurements on each component. We can provide the data in different formats. As I mentioned, the digital radiographs are the raw images. After reconstruction, we get the kind of slice image I show on the left, the top row, which is basically a slice image of a soil core. Upon reconstruction, the colors invert. So here, the lowest density component, the air, will be black, darkest, and the highest density component will be the brightest, white. So from the reconstruction, we can provide this kind of image slices, image stacks, but we can also do 3D rendering of volumes, such as the middle, where I show a clipped portion of the 3D rendering of a soil core with the porosity colored in blue. And we can also visualize the pore connectivity on the right. I'm showing the connected versus unconnected porosity in the same soil core. So as you can see, we can color components, make one transparent, introduce clipping planes to kind of highlight, emphasize components that we want to see. And in terms of file formats, so the 3D data is in most cases .vol or .row file format. It can be opened with using different software. The image stacks we can provide in TIFF or other JPEG or any image format, and we can create animations, AVI files, just slicing through or more sophisticated animations. Some further examples are shown in this slide. On the right is a lily flower. Also, you can see the 3D rendering versus the one slice image and the false coloring, or the segmented root image at the bottom. So a few words about segmentation. So a typical workflow for segmentation would be like this. So after reconstruction of the 3D data, we typically perform some filtering and enhancement of the images. That depends on the target component, in this case, the root. So this example is actually a plant soil system, which even contains an added 3D printed grid inside the pot. This was used to kind of create compartments within the sample for the root and soil. So if we are after the root here, we would need to do the filtering in a way that we try to enhance the contrast for the root. And then the thresholding would be the next step, where if we have a large enough contrast between the components, then we can use an automated thresholding. This is basically where we distinguish the different components and attribute different intensities or colors to them. In most cases, in the soil systems, it's a challenge. So it becomes an interactive process or even manual, moving from slice to slice and paint it carefully. And then once we achieve a segmentation, like the one on the right, we might have some particle-like features floating around, basically leftover phases of the same density as the root. We can filter them out by size or using an erosion operation as needed, and then clean up the image. And then on the final image, we can do volumetric measurements such as volume, surface area, etc. So a few challenges that we are working on and trying to address. One is the phase component contrast, as I mentioned. This is the same example as on the last slide. I highlighted or zoomed in on an area where you can see how the root piece is of the same color or density as the plastic container and the polymer grid. So you can change, if you can, the sample containment or the density of the grid, or maybe try different soil if the project has that flexibility. But if not, then you have to use a segmentation approach that can address these issues. And so often this is manual segmentation. if you run an automated like watershed segmentation, you will find that it's not sufficient, and often you end up doing a kind of a sequence of operations, such as using one segmentation and then another for another component, and maybe subtract images from each other, kind of in an iterative way until you get a clean image of the desired component. Another challenge is the sample size versus resolution issue, where we all would like high resolution images on the whole volume, a large sample, which is a contradiction. Often we need to zoom in or subsample and a sample to get higher resolution data. One way to go around it is to, if you have a cylindrical shaped sample, you might be able to scan it in several tiles, going up, down, and we can stitch the images into a full data set at higher resolution. Data size is another challenge. Typically, we end up getting tens of gigabytes of data from one scan, which can be an issue if somebody wants to view the data on the laptop. So often we need to reduce the sample, the data size, or the bit size, which is 32-bit originally, but maybe you can only work with 8-bit data on your laptop. The other thing we work on is automation. So we are interested in automating both the data collection step by being able to scan multiple samples in a sample changer, and then also to automate our our segmentation workflow by AI and machine learning tools. In this example here at the bottom, we are comparing our manual segmentation achieved in a rhizo on this soil aggregate with that achieved in using a machine learning code of one of our collaborators. And as you can see, after sufficient teaching of the algorithm, we actually can get better segmentation of the components. And so this is very promising that we will try to implement on high performance computing for users. And with that, I'd like to give the stage to Pubudu, who will also mention a specific example where we collaborated on XCT, and she used for her project. Thank you.
Thank you, Tamas. So I am Pubudu Handakumbura. I am a plant biologist and a team lead at EMSL. And today I'm going to share an example, a case study where we have used XCT and some of the other analytical platforms at EMSL to visualize the root rhizosphere zone. So as many of you might know, rhizosphere is one of the most dynamic interfaces on Earth. And this fosters numerous inter- and intra-kingdom interactions. And we really want to understand the system as a whole in a near native environment to really get a predictive understanding of the system so we can better accommodate future ecosystem designs. But visualizing this hidden half is a grand challenge. So I think the 3D root cartography platform we have developed here at EMSL is complementary to some of the other techniques that are out there. Next slide. So this is the workflow of the 3D root cartography platform. So for this, we have used 3D printed rhizogrids, which are illustrated on the right-hand side of this workflow. So by using these rhizogrids, we are able to provide a scaffold to the plants, and then that helps us keep the root system in place as we are going through all these various steps. So the first step of this workflow is growing these plants in these 3D printed rhizogrid fitted 3D pots, and then imaging them using XCT. Once we have captured all the images, then we are able to use the same samples for other destructive sampling methods. So in this example, we have used LCMS metabolomics platform and 16S sequencing for looking at the root exuded metabolites and the microbial abundance along the root rhizosphere zones. And because we have these rhizo grids, that allows us to have a 3D coordinate along the roots within this pod. So that helps us to bring all of this different molecular level information back to the root pieces to map them back onto a reconstructed root system, which is illustrated here at the bottom with the different colored molecular information mapped. So if we go to the next slide, we can, so this is a real example where I have used these rhizogrids, and I used a field soil for this example, so from a local field site. And we have used sorghum for this particular experiment. This is a 5-week-old sorghum plant grown on these rhizogrids. And as you can see from this aerial view, there are 4 quadrants to each of these rhizogrids. We've marked them A through D. That helps us provide a 3D coordinate for each of these root segments that we segment later on. And then the bottom figure is a XCT image, and the image on the right is the reconstructed roots and the rhizogrid from this experiment. If we move to the next slide, this is a 3D illustration video of this same sorghum root system. So now, since we have captured the root system in a near native environment, and we have preserved this root system architecture, then we are able to use this information to say something about the root physiology itself, the branching architecture. We can extract different information from these XCT images. And taking this one step forward, in the next slide, as I've mentioned before, so we have coupled this approach with molecular phenotyping. So now, after capturing the XCT images, we are sampling these roots and the rhizosphere. And so this, in this specific example, even though I have used the LCMS platform that's available to us at EMSL and the 16S sequencing, we can use different sampling approaches here. It could be proteomics, it could be transcriptomics. So various analytical platforms can be coupled to this, and that information can be mapped back to the roots. So in the next slide, this is an example of 5 selected metabolites that we are illustrating the abundance, relative abundance through the same root system. So you can see on your left, the aspartate seems to be relatively highly abundant towards the crown of the root, whereas the phenylalanine, for example, that's shown in the middle, seems to be low abundant throughout the root. So you start to see the differences between these metabolites along the root system. And in the next slide, this is another example. Now we are again using the same root system and the information extracted from the same rhizosphere and the roots. And here we are looking at the abundance of 5 different microbial genera. And again, you see that they are not uniformly distributed along the roots. So this allows us to have some spatial resolution for our root exuded metabolites, as well as the microbial genera that are colonizing these roots. So with that, I want to acknowledge everyone who's in this slide. And this was part of the persistence control of engineered functions in complex soil microbiomes, SFA, that was recently awarded to PNNL. And part of this work was done at EMSL. And with that, I'm happy to take any questions. Thank you for your time.
All right. Hi, everyone. So we're going to move on to the Q&A portion of this webinar. So let's see. We've been capturing all the questions you've been putting in the chat window. So let's start first with Amir. So will the same look at NARI be featured next week during the Integration 2021 meeting?
Yeah, actually, so next week integration, we have lots of things to show, and I really encourage everyone to attend. I'm not sure if NARI is a significant part of that, but it will be probably mentioned as part of one of the tutorials. But there are many other exciting things, and I'm happy to share more information about NARI even during integration with private chat or any way that works with users and audience.
All right. So the next one for Tamas, how much human interaction is needed to get a sample in the system for imaging?
Well, currently, we have to put it in physically. So the user, the operator would need to lean into the cabinet and put the sample on the stage. And also, depending on the size and nature of the sample, you might need to pick a different sample holder. So a cylindrical container or pot can be placed on a flat surface, but when we have a smaller sample, we might put it in a tube, a cylinder, or at the tip of a tweezer-like holder. And what we are trying to develop is the ability to. We have a concept now where we would have multiple soil pots or soil tubers that could be loaded into a stage, and then either a robotic arm would lift them into position, or the sample would, or the stage would translate horizontally and move to the next sample. And so we're working on that design.
All right. And for Pubudu, can you delve into how 3D printed rhizo grids are made?
So we have a machine shop in the lab. So we have instrument staff who are capable of 3D printing these. So this was with a lot of trial and error that we settled into this setup. But if you have access to a 3D printer, this is much easier. But we did, like Tamas mentioned, we did have to test density of the grids, density of the growth medium we are using. So we had to test on all of those and settle in on a diameter that works well, that does not compromise the root system architecture. So I think as to the specifics, so it would be something that we could chat offline, but that these can be customized. So that's the nice thing about it. So you can customize these according to the species you're studying or the project that you're working on.
All right. So the next one for Amir, I'm sort of rotating through these questions, so they're not in order that they were in the chat window. So for Amir, does the transparent soil present challenges of light contamination? And if so, how do you correct for that?
Yeah, it's a great question. So what we usually do, we actually cover the jars just with foil, just to reduce that light contamination. And our system, as I mentioned, has that NIR LED backlight, which actually reduce that contamination during imaging. So compared to the visible light, these are our solution for that. But we haven't really seen a very significant problem there so far. But yeah, covering the jar with something like a foil would be our solution for now. But it can be other solutions to even make it better.
All right. So the next one for Tamas, how close is the imaging system to being able to queue up a set of samples and allow the system to image them one at a time?
Yeah, this is what I was hinted at before. We don't have that stage built, but we have a design. So the problem there, or the challenge there, is to, once you have a sample mover or either a robotic arm or whatever, it has to be integrated into the instrument software, because, as you know, you control the scanner using a program where you would, after setting up the scan conditions, you would do a calibration, we call it shading correction, and then start the scan with the sample in place. And so the sample changer has to be integrated with this workflow, meaning that one scan is done, and then it would move the next sample onto the rotation stage, because the sample has to freely rotate, and then the software, the instrument software, has to know that, acknowledge that, and once it's in place, it will start the new measurement. So it has to be a programming component, like a scripting or coding component to it, and I think we have the right interface now that we can use for that.
All right. And Pubudu, I think this one was for you. So thank you for the excellent presentation. Are the imaging data open to share?
So they will be. So some of this is published as an opinion paper at M Systems. We are working on a manuscript currently that describes the data, because there's a little bit more data than what I've shown. But I think, yes, that can be shared. But this would be large files. So we would need to figure out like how to allow access to those files, but yes.
All right. Let's see. Sorry, there's lots of questions coming in now. Actually, I'm going to do one more for Pubudu. Were the metabolite samples taken in situ or after destructive removal? And is it possible to monitor metabolites in a growing root system over time?
So that's a great question. There are limitations to what all these methods that we proposed and are out there. So what we have right now is from destructive sampling. So we have sacrificed the sample we have used for imaging to then do the downstream molecular phenotyping. So right now we don't have a way of continuous monitoring other than XCT, because if we want to capture the root at the stage where we have done imaging, we are doing destructive sampling, because we have allowed these to grow in almost near native conditions, so in soil. So there can be, like with different probes, that would be something. I know there are parallel efforts of using optodes and different types of probes. So that would be one way of integrating maybe some of those for continuous monitoring. But what we have right now, what I have shared with you today, does not allow us for continuous monitoring. But you are able to, grow a lot of individuals and sample them across development. So that is doable, but each sample is sacrificed at the end of imaging.
All right. So for Amir, NARI measures so many root traits. How do you link root traits with root function?
Yeah, that's a really good question. And really that was one of the major goal of developing this type of phenotyping facilities, because you really eventually want to relate those root phenotypes to some functions or some genotypic information. So the thing is that the system by itself can only, obviously can only provide data with root phenotypes, plenty of root traits. it depends on how we really design the experiment, and depending of the goal of that individual studies. For example, if we have some mapping populations, that we can use this root phenotyping approach to find some QTLs or quantitative trait loci. That's a way that many people use and combine this genomic information with root phenotypes. Or if we have some particular mutants deficient for specific genes related to root growth, then we can also use those selections of mutants and grow them in our system and image them, and then try to correlate those information with the molecular data. Yeah, it all depends on how we really want to design the experiment and what type of species and populations we have.
Okay. Looks like we've got just a few more minutes. So Tamas, what is the minimal density difference for substances to be distinguished by the X-ray CT? Mm-hmm. I know that you answered it in the chat, but it actually only went to the panel, your answer.
Oh, okay.
So that's why
no problem. Yeah. So this depends on the sample and of course the different component, but usually the sample overall, and of course the X-ray settings and the scan settings. I can give you an example. We are successfully differentiated pore water inside pores. You know, the air and water has quite different density, but in the CT images, because the water has only hydrogen and oxygen atoms, it doesn't come across as a high density component. So air and water is always difficult to distinguish in a soil core. But when we collected better data, long scan on a small sample aggregate, then we could distinguish those components. So we cannot talk about a specific difference value, but density value, but it can be investigated by changing the settings. Definitely, the smaller the density difference, the better signal to noise ratio you need. So the longer you collect, but better data statistics always helps.
All right. Let's see. So Pubudu, let's see, do we have any? No, let me just throw out a generalized one. If any study with different pH environments around the root, I would like to know, as I propose to have a vivo studies for a new DTWRS spectrometry with varying pH microenvironment in specific to molecular phenotyping. You would like to know if you I'm not quite sure where the question is on that. I'm sorry if you were the one to ask this. Maybe if you could clarify in the questions. Let me go to another one. What is the typical time frame needed to perform a root growth imaging experiment with your model plants? Anybody can answer.
So I guess it starts with the growth, right? So maybe the plant scientist with the growth.
Yeah. So what was the question again?
Well, it's going to have to be short because we're out of time. What is the typical time frame needed to perform a root growth imaging experiment with your model plants?
Yeah, with NARI system, for one individual plants, just take about 10 minutes to get all the images. And then by that, we can possibly image about 40 to 50 plants per day. But regarding the duration or the developmental stage that we can image, so with NARI, so we really cannot keep the plants for a long time in those jars. So we usually capture the early developmental stages. So I hope that answers the question.
Yeah, we're out of time. So I just want to let everyone know, I know there was a lot of questions we didn't get to. In the chat window are the email addresses for Tamas, Amir, and Pubudu. If you have more questions you want to ask, please feel free to email them. directly, and they can answer your questions. So with that, thank you everyone for a great webinar. We appreciate that you're here, and have a great day. We hope to see you next month.
Thank you. Bye.
Thank you.
Thanks.